Hello everyone, welcome to my channel. Today's topic is infrared spectroscopy. What is IR spectroscopy? IR spectroscopy is a technique used to identify chemical substances or functional groups by analyzing the interaction of infrared light with matter. How does it work? Infrared light is passed through a sample. Different molecules absorb specific wavelengths of infrared light, causing their bonds to vibrate. The absorbed light is detected and recorded, producing an infrared spectrum. The spectrum shows peaks corresponding to different functional groups within the molecule. What does it tell us? Identifies functional groups by analyzing the positions of the peaks in the spectrum. Chemists can determine the types of bonds present in a molecule. Determines molecular structure. The overall shape of the spectrum provides information about the molecule's overall structure. Analyzes purity. IR spectroscopy can be used to check the purity of a sample by comparing its spectrum to that of a known pure compound. Common applications of IR spectroscopy: organic chemistry, functional group identification. IR spectroscopy is invaluable in determining the presence of various functional groups, like carbonyl, hydroxyl, amine, etc., in organic compounds. Structure elucidation. By analyzing the positions and intensities of absorption bands, chemists can deduce information about the molecular structure. Isomer differentiation. IR spectra can help distinguish between isomers, as they often exhibit different absorption patterns. Pharmaceutical industry, quality control. IR spectroscopy is extensively used to ensure the purity and consistency of drug products. Drug development. It aids in characterizing new drug compounds and monitoring synthetic processes. Polymer analysis. Polymers used in drug delivery systems can be analyzed for their composition and structure. Environmental science. Pollution monitoring. IR spectroscopy is employed to detect and quantify pollutants in air, water, and soil. Climate studies. It helps in measuring greenhouse gases like carbon dioxide and methane. Food industry, quality control. IR spectroscopy can be used to assess food composition, detect adulteration, and monitor food spoilage. Food safety. It can identify contaminants and allergens in food products. Forensic science, drug identification. IR spectroscopy is a crucial tool in identifying illicit drugs and their metabolites. Polymer analysis. It can help analyze synthetic fibers and plastics found at crime scenes. Explosives detection. IR spectroscopy can be used to identify explosive materials. Components of an IR spectrometer. Radiation source. Emits a broad spectrum of infrared radiation. Common sources include glowbar, silicon carbide, Nernst glower, rare earth oxides, nichrome wire. Sample holder, holds the sample to be analyzed. Can be in various forms depending on the sample state: solid, liquid, gas. Common types include liquid cells, solid sample holders, e.g., KBr pellets, ATR crystals. Gas cells. Monochromator separates the infrared radiation into its component wavelengths. Essential for dispersive IR spectrometers. Uses prisms or diffraction gratings to disperse the light. Detector converts the infrared radiation into an electrical signal. Common types include thermocouples, bolometers, pyroelectric detectors, recorder or computer, displays and records the infrared spectrum. Modern spectrometers use computers to process and analyze the data. Key steps in spectrum interpretation. Identify the functional groups. Region 4000 to 2500 cm superscript 1. Focus on peaks associated with OH, NH, and CH stretching vibrations. Region 2500 to 2000 cm superscript 1. Look for peaks indicating triple bonds like C is identical to N and C is identical to C. Region 2000 to 1500 cm superscript 1. Identify double bonds, particularly C equals O, C equals C, and aromatic C equals C, region 1500 to 650 cm superscript 1, the fingerprint region, complex with overlapping peaks, is useful for confirming functional groups and differentiating compounds. Analyze. Peak shapes and intensities. Broad peaks often indicate OH or NH groups. Sharp peaks, typically associated with C equals O, C equals C, and C is identical to C bonds. Strong intensities usually correspond to polar bonds. Weak intensities may indicate less polar bonds or overtones. Consider the chemical context. Combine information from the IR spectrum with other analytical data, e.g., NMR, mass spectrometry, to confirm the structure. Consider the possible compounds based on the given information or experimental context. Use reference spectra. Compare the unknown spectrum with known spectra of similar compounds to aid in identification. Spectral databases can be valuable resources for this. Purpose. Operating procedure. Sample preparation. Solid samples can be prepared using several methods. KBR pellets. Grind the solid sample with potassium bromide and press into a pellet. Mole method. Mix the solid sample with a mulling agent like mineral oil to form a paste, then place between two salt plates. ATR, attenuated total reflectance. Place the solid sample directly on the ATR crystal for measurement. Liquid samples. Place a small drop of the liquid between two salt plates or use an ATR accessory. Gas samples. Use a gas cell with windows transparent to IR radiation and fill it with the gas sample. Advantages of IR spectroscopy. Functional group identification. IR is excellent at identifying functional groups within a molecule based on characteristic absorption bands. Qualitative analysis. It can be used to identify unknown compounds by comparing their spectra to reference libraries. Non-destructive. The sample is generally not consumed or altered during the analysis. Versatility. Applicable to a wide range of samples, including solids, liquids, and gases. Rapid analysis. Modern FTAR. Spectrometers provide fast data acquisition. Quantitative analysis can be used to determine the concentration of components in a mixture, complementary to other techniques, often used in conjunction with other analytical methods for comprehensive analysis. Limitations of IR spectroscopy. Complex spectra, overlapping peaks can sometimes make interpretation difficult. Limited structural information. While it provides information about functional groups, it doesn't directly determine the complete molecular structure. Sensitivity. Less sensitive than some other spectroscopic techniques for trace analysis. Water interference. Water absorbs strongly in the IR region, which can complicate analysis of aqueous solutions. Sample preparation. Some sample preparation techniques can be time-consuming. Matrix effects. The presence of other components in a sample can interfere with the analysis. Infrared spectroscopy is a powerful and versatile analytical technique essential in various scientific and industrial fields. Its ability to provide detailed information about molecular structure and composition makes it invaluable for research, quality control, and environmental monitoring. Thank you for watching. Do not forget to like, share and subscribe.